Hello everyone. In our today's lecture of microbiology part first, we are going to learn about the contribution of scientist Martinus William Beijerinck in the field of microbiology. Now, Martinus Beijerinck was a Dutch microbiologist and botanist. He is often considered as one of the founder of virology and environmental microbiology. In spite of his numerous pioneering and influential contribution to the science in general, he was never awarded with the any of the award. Beijerinck discovered nitrogen fixation and the process by which diatomic nitrogen gas is converted to ammonium ions and becomes available to the plant. Bacteria perform nitrogen fixation by lodging inside the root nodules. Now, different types of nodules or legumes are formed at the roots of plants in which the nitrogen fixation or nitrogen is fixed. Now, you might have seen various nodule producing plants like small bird like structures are produced on the plants which are helpful in the proper nitrogen fixation. He isolated a Bacillus rudicola species and proved that it forms a nodule on the root of leguminous species. Later isolated Rhizobium species and studied the process of nitrogen fixation. And he also performed and discovered various biochemical reactions which increases the soil fertility and their use in the field of agriculture. Then he also contributed in the various scientific researches like in the discovery of the phenomenon of bacterial sulfate reduction, a form of anaerobic respiration. Now, the anaerobic respiration is nothing but the microorganisms which grows in the absence of oxygen, or respiration process which is carried out in the absence of oxygen is known as anaerobic respiration. He learned bacteria could use the sulfate as a terminal electron acceptor instead of oxygen. When he was studying during his research work, he found that not all the organisms required oxygen for the respiration process. Some bacteria or some organisms can also use sulfate as the terminal electron acceptor and does not use oxygen, and he termed this process as anaerobic respiration, and the organisms which perform their respiration anaerobically were known as anaerobes. This discovery has an important impact on our current understanding of biogeochemical cycles. Spirulum desulfofuricans, and now this spirulum desulfofuricans is known as desulfovibrio desulfuricans. This is the species which is the first known sulfate reducing bacterium, or the first bacteria which was known to use sulfate as a terminal electron acceptor. was isolated and described by the scientist Beijerinck. Beijerinck invented the enrichment culture, a fundamental method of studying microbes from the environment. Now, enrichment culture is the use of certain growth of media to favor the growth of particular microorganism, or the supply. Enrichment culture is nothing but the media or certain material which contains the proper nutrient for the growth of microorganisms, or the compound which favors the growth of microorganism is known as enrichment culture. This is generally done by introducing nutrients or the environmental conditions that only allows the growth of an organism of interest, the microorganisms which we want to grow. Only that microorganism can be used or can be produced by using enrichment culture. This was all about the contributions of scientist Beijerinck, scientist Martinus William Beijerinck in the field of microbiology. He discovered the nitrogen fixation process. Also, he discovered the anaerobic respiration process and developed the enrichment culture for the production of different types of microorganisms. Thank you.